Hello, I'm Llewellyn King, the host of Any CFS Alert. Today we have a very special broadcast with a very interesting and unusual woman. She is a pure researcher with a great history of research, not in myalgic encephalomyelitis or associated disease, but I thought it would be interesting to see where research across the board stands and what the fallout, the constructive fallout, the collateral effects, if you will, of COVID-19 are in research generally, and what hope there is for other diseases, and of course, especially for ME. Linda is the Chief Executive Officer. Her name is Linda Marban, I should have said that. She's Chief Executive Officer of Capricor Therapeutics in Los Angeles, and she has had a most extraordinary career. Linda, would you like to tell us the high points of that career?
Thank you so much, Llewellyn. I feel eternally grateful for the career path that I've had. I started off life actually as an elementary school teacher, and then decided that I was called to science. It was sort of a confluence of curiosity and also the desire to learn more about medicine and the idea of making people's lives better or longer. I started on a trajectory where I was living in Ohio, where I went to graduate school. I raised 4 children and went to graduate school, barely surviving on a graduate student stipend of $19,000 per year. And I actually still tell my children the story of taking my physiology exam the morning after my children had been up all night with the stomach flu, barely being able to drag myself in. So graduate school was a long journey with 4 small children. And then I was lucky enough to get a postdoctoral fellowship at Johns Hopkins University in the laboratory of Linda Marban, who had been a leader in the space and someone whose work I had been following since my introduction to science. He was a cardiovascular physiologist of the highest regard, and so I was lucky enough to secure a position in his lab as a postdoctoral fellow. Moved on to junior faculty at Johns Hopkins, where I studied cardiac muscle physiology, so basically what causes heart disease and how we could potentially fix heart disease. But I always had the bug to go into research, so very early into my tenure academically, I was recruited to work in a gene therapy startup that was working on gene therapies for cardiac arrhythmias out of Johns Hopkins University called Exosomes. And this company was funded by a company called Genzyme. And so I said I went to biotech boot camp. I basically learned how to build a startup company by learning from those that were already actively involved in business development and also product development at Genzyme. And then somewhere along the journey in that early stage gene therapy company where gene therapy was really having trouble getting launched, we couldn't figure out how to get genes into those viral vectors in those days and how to get them to express, and the regulatory pathway was long and tortuous. We came across the technology that became the foundation of Capricor in the Isle of Capri, hence the name Capricor, and we found these cells that seemed to have healing properties in terms of managing cardiac dysfunction. So in these little mice that we treated with the cells, we were able to see that if they had experimental heart attacks, the heart attacks got smaller and better. The Italian scientist, her name was Elisa Messina, who had done this groundbreaking work, then came to Johns Hopkins, where she spent over a year perfecting it, along with somebody who became one of the foundational employees of Capricor, Rachel Ruckteschell-Smith. And the combination of those women led to the founding of the company Capricor. I helped to found the company, bring in the intellectual property from University of Rome at La Sapienza and Johns Hopkins University, founded the company and began to build it back in the days when we thought the cells that we had were a stem cell, and the stem cells would then go in and fix broken tissue, so to speak. What we learned along the way is that the cells don't work by stemness. Our cells work by releasing exosomes, which are nanometer-sized lipid bilayer vesicles that mediate cellular communication and drive healing through changes in protein expression, protein translation. So that's sort of the 5-minute elevator version of how I got to where I am today.
Well, it's pretty remarkable. And Capricor is working on an inoculation against COVID-19, and also a treatment, a therapy for use in the hospital, which is the repurposing of a drug that you've developed for muscular dystrophy. Tell us about those two things, and also a little bit more about Capricorn. How big is it? Is it, tiers of white-coated people staring into microscopes and Petri dishes full of strange things happening? How is it done? What's it like?
Capricor has about 30 employees. Our market cap is hovering right now around $100 million. We've been around for a little more than a decade, and we've moved through sort of the torturous waters of cell therapy. I spoke a few moments ago that when we founded the company, we thought that we were developing a stem cell therapy for heart disease. We had the nimbleness and the ability to move quickly as we understood the science better, and we moved our products forward. So we moved quickly off the concept of stem cell therapy to the idea of cell therapy, using our cells to combat injuries to tissues. And what we learned about our cells is that they are profoundly immunomodulatory. That means they help balance out the immune system. And so really, anti-inflammatories have certainly their place in therapeutics. We've all taken Motrin for a headache. But in reality, when you have muscle injuries or injuries to certain tissues, you don't necessarily want to knock down all inflammation. You want to knock down bad inflammation and encourage then those pathways that stimulate repair. So believe it or not, in your body, certain inflammatory pathways stimulate repair. Our cells slow down the bad stuff and speed up the good stuff. So how did that take us from Duchenne muscular dystrophy to COVID-19? Well, Duchenne muscular dystrophy is a genetic disorder where primarily boys and young men, it's over 90% of the patients are boys and young men. It's because it comes on the X chromosome. It's called X-linked, and it's typically from their moms. And what you see then is that they don't make dystrophin, which is the largest protein in the body, and the cells can get injured very easily and break down. And when cells break down, the immune system sees that something bad is happening. So that stimulates all kinds of inflammation. So these patients are battling two phenomena all the same time. One, the lack of dystrophin leads to cellular injury, and two, the breakdown of the cells leads to inflammation. So our cells, CAP-1002, are able to manage both sides of that coin. They're able to calm down the inflammatory pathways, stimulate endogenous repair, and then support new muscle development, which is why we've seen such positive data in our clinical trials for Duchenne muscular dystrophy. Well, okay, so you're thinking, okay, but COVID-19 is a virus. A virus is killing people. What does that have to do with any of this? Well, really what's killing people and making them so very sick is not just the virus. It's the sequelae that comes. It's the cytokine storm, we call it, or the hyperinflammatory syndrome that occurs afterwards. So basically, if you can think about it this way, the virus goes in and weakens the body, and the immune system starts fighting, and they fight hard. And sometimes, even as the virus abates and it goes away, the immune system has been revved up so much it can't calm down. And that's what stimulates these terrible things like what we call ARDS, which is acute respiratory distress syndrome, which is the pneumonia that you typically see with COVID. Our cells are very good at dealing with those inflammatory consequences. Again, harking back to the mechanism of action. Calm down bad inflammation, stimulate good immune system responses so that we have healing occurring. And what we're going to be doing then is, in our future efforts in COVID, is targeting those patients that are at greatest risk. So those that are coming out the backside of the virus but aren't fully in cytokine storm yet. So we have a chance of driving them back sort of from that colloquial cliff of morbidity and mortality.
In chronic fatigue syndrome, also known as myalgic encephalomyelitis, which I've taken an interest in for 10 years now because friends are afflicted, one of the concerns is there isn't enough research, i.e. not enough money. I wonder what you do if you get the money. Is the talent there to tackle other diseases when so much of the talent seems to go to the big diseases, heart, diabetes, cancer, obviously cancer, and everybody who suffers from or sponsors or cares about a particular disease would like to see more research. How do you feel about the way we manage research in the US? We raise money and we send it off, and we tend to start thinking that it's all about money, but it's really all about science and talent, isn't it?
Well, science and talent takes money. So this is a conundrum that you've raised, Llewellyn, which is one that causes great consternation to those of us that are deep in the space. It takes money to build therapeutics, whether it be from an academic lab or in a corporate lab. But in a corporate lab, what we have to do is we have to continually fight for the right to stay alive by raising money. And the way that we do that is we establish milestones. And you have to hit your milestones so that you can get more money to keep moving forward. Unfortunately, in diseases like chronic fatigue syndrome, it's very hard to do one of the most important milestones, which are clinical trials. So it is actually probably the worst case scenario for those poor people that suffer from this, because it's a disease without a clear projective pathway, not clear symptoms, not clear trajectory, and not terminal. So you have a situation where you can't build a clinical trial that can be done in some reasonable amount of time with a reasonable number of people to develop therapeutics specifically for something like that. So it falls by the wayside. And there's also, let's face it, a lot of hand-wringing that goes on when you have something that doesn't have specifically a biomarker. So I haven't studied chronic fatigue syndrome in a long time, but I had a great interest in it a long time ago. And unless things have moved specifically, there was really no way to measure it. So it was sort of cast off as a sidebar to depression or something head scratching or something related to, a mental illness pathway, as opposed to something that was physiologic. And yet these poor people that are impacted by it are suffering terribly physically. And so we don't have a good meeting of the minds in terms of developing therapies for chronic fatigue, as I'm aware of today.
Let me go back to when you were explaining what Capricor does and the vaccine. How are you coming with the vaccine? And as there are 100 vaccines under development, how will one of them get out in front, assuming that they all work, or that half of them work, or that 10% of them work?
So it's a very competitive space. Of course, you've taken every scientific mind literally worldwide to work on this. So why would a baby company with a $100 million market cap like Capricor want to work on this? Well, it's because we believe, and our science team believes, that we have a way of building this vaccine that could be much more beneficial than what's currently being used. So basically, everybody in the space is working on several opportunities, and they're all slightly different variants of either a live attenuated vaccine, a killed vaccine, a killed viral vaccine, or a recombinant vaccine, or a virus-like particle vaccine. And we are working on both the recombinant and the virus-like particle vaccine. Now, why would we work on this when Pfizer's working on it and Moderna's working on it and some of the other big pharma companies are actively working on these technologies? Because we have something called an exosome. So if you remember a little bit ago, I told you that along our scientific journey, we discovered that the mechanism of action of our cells are through these exosomes. And exosomes are actually the words of cells. They are tiny little particles, nanometer-sized lipid bilayer vesicles. They're a millionth of a millimeter that were discovered in the 90s and considered to be the trash can of the cell. And then somewhere about 10 years later, they were opened up and somebody looked inside, and the prescient scientist said, this is not trash. This material inside these exosomes are actually for communication. And so it opened up a really now 15-year journey into understanding the exosomes. And what they are nature's perfect delivery vehicle. So for 30 or more years, we've been looking at ways to deliver technologies or products or proteins to the nucleus. And now using the exosomes, we can do this. We can get across the cell membrane all the way through the cell and then change how a cell functions by using these exosomes. So we realize that we'll be a second generation vaccine candidate. We are excited about that. It's very unlikely that almost anything out there will work perfectly and certainly won't work in the short term. And we believe if the data continues to bear fruit the way it currently is, that we have a way of building this vaccine that's not only unique, but more effective.
Linda, how many diseases do you think might be helped by collateral discoveries while chasing after a cure or a therapy for COVID-19?
It's interesting that you asked that because it's one of the things of great discussion. It should really change vaccine development for all time. Hopefully it'll change how flu vaccines are made. We should be able to make an Ebola vaccine, maybe even, the hardest of all, an HIV vaccine, because we should now learn how to actually do it, how to make them in large quantities, and then how to quantify their effectiveness. So vaccines have sort of been the poor sister in the development of technologies for a very long time. Nobody cares very much about it. There are companies out there that were barely staying alive trying to develop flu vaccines that now have market caps that are in the hundreds of millions or billions of dollars because we all of a sudden, as a society, realize how important it is to be able to protect ourselves against the pathogen. Tiny little virus can cause so much death and destruction.
And how do you work? When you're looking at cells, do you look through electron microscopes? Do you look through optical microscopes? Do you squint? I mean, this is all a mystery to those of us who are not in the world of cells.
So we are scientists, I always tell my students, are professional sleuthers. We're detectives. And what we do is we take advantage of the armamentarium that we are offered, which is that we can take signals like light, like sound, like physical properties and turn them into ways of measuring something that is too small to see, but then something that we can then measure, like, for instance, emitted light is a wonderful way, and that's how we can kind of quantify whether somebody has a virus. PCR, everybody's now talking about a PCR, a polymerase chain reaction. It's how we're measuring whether we have COVID or don't have COVID. It basically takes a little tiny section of a gene and then amplifies it up many, many times until you can see it. And then when you see it, if it's there in your sample, you can measure it, and then you know the patient has COVID. So we use a lot of things in the lab. We use microscopes, as you said, optical microscopes. We use electron microscopes if it's something that's too small to see optically. We use light, and we use sound, and we use all kinds of imaging technologies in order to be able to track where things are going or how they're functioning. And then we use straight-on, beautiful physiology, and I'm a physiologist by training, so it's my sweet spot, but we look at whether a mouse can run farther or longer on a treadmill. For instance, in our Duchenne Muscular Dystrophy program, that's how we learned that our cells were working. Our mice could run. Or we look for their ability to mount an antibody response. We can take a little bit of blood and we can look for it, whether it lights up. If we have a little bit of light, then we have a little bit of antibody. If we have a lot of light, we have a lot of antibody. And so that's how the trajectory is. It goes from very small observations in Petri dish or test tubes all the way through animals where we can safely test whether or not we're going to see the same thing we're going to see in you and me.
When do you think you'll have be positive about your vaccine?
Well, as I just mentioned, it's graded steps along the way. The first step is to look as to whether our vaccine candidates will make antibodies. We're pretty confident that's going to happen, and that's coming very soon. Then we have to do a viral challenge. So you put them typically into non-human primates at the vaccine, you vaccinate them, and then you expose them to the virus.
That's the poor old monkeys.
That's right. We use monkeys. We don't do it. We have a very special facility that does it. And then they are exposed to the virus. And if they don't get it, then you can declare a success. And then you go into humans. If all things go well, we expect to be in humans with our vaccine candidate in the early to mid part of 2021.
What about off-label uses, what are called off-label uses, where for so many things, including your muscular dystrophy work, where things designed for one purpose have a wonderful effect in another place if you know to try them, or if you even know that they exist. Now that should get better as we are better at managing data. The hundreds of thousands of compounds that have been put together over the decades to make medicines, some of them may have uses that we have no idea of. Does that concern you? Do you get into that at all? And how are we going to use those? How are we going to find a way of finding medicines that exist and repurposing them?
Well, it's a multi-pronged answer to a very complicated question. So many wonderful compounds are left on the shelf because they fail in clinical trial. And we've built this system really over the past 50 years of clinical trialing, where we really have to put our chips on the black or the red circle and hope that the therapeutic delivers what we've asked it to in the way that we think it should in order to take it forward for a certain indication. If it fails there, it doesn't mean it's a failure as a drug. It means it didn't meet that endpoint. So our shelves, not ours at Capricor, but our shelves in sort of the colloquial world of drug development are filled with these compounds that work, but we haven't really elucidated how to measure that they work. In terms of off-label uses, those compounds that are on the shelf aren't usually being used for that because they're not approved yet. If they've been approved, that's when off-label use comes into play. And generally, we typically know the mechanism of action at that point, and then it's sort of matching those puzzle pieces together. You know, I've got an anti-inflammatory that works in spinal cord injury, and now I've got a muscle that has inflammation going on. Maybe this will work, and you can try it and see if it does work, and then a physician has a way of prescribing it. It has economic impact because we don't know how to quantify how much to charge for that or how to qualify whether it can be actually used for that purpose. So then we have to go back into the clinic to do another clinical trial to get that specific indication on the label.
That's fascinating. Are we going to see, because of mega data, because of our ability to manage data, are we going to see better uses of what we've already discovered?
Yes, I think that's very, very true and very likely, and it's already starting to happen, where compounds can be pulled down off the shelf and used and redeployed for many different uses. And I think that our treatment of disease will become more targeted and more successful. Now, I am a huge believer, obviously. I run a company that's developing an exosomal-based platform therapeutic. I think exosomes will change biotechnology the way that antibody therapy did. Exosomes are perfectly suited as nature's communication device or delivery vehicle to be packed with different molecules for repair, for healing, for changing gene expression, for pretty much anything you can think of. And as we begin to harness their power, I think we will be able to have more directed therapeutics that have fewer side effects as we can direct them exactly to where we need them, as opposed to, for instance, taking something to kill the pain in our knee, but because we have to deal with all of the pain receptors in the body, we end up feeling groggy and tired and non-functional. So hopefully we'll be able to direct painkillers directly to the knee and still be able to have pain relief and function.
Let's go back to the two non-scientific issues, money and talent. How does your company get its money? It's an investor-owned company, so presumably it's always got the money in the market. It doesn't work on government grants, right?
So we've gotten a lot of government grants over time. It's how we really built the company. So as I mentioned, we were sort of founded and did our toddler and early childhood development in stem cell therapy space where nobody was putting any money. We were very lucky to have some brilliant people working with us at the time that were able to write grants. And so I think, and I could be wrong on this, so you have to check our filings, but I think we've gotten about $60 million in grant money over the last, let's call it 5 to 10 years. But we also now are publicly traded, and so, we typically raise money from investor base, just like anybody else that's public, sell stock and ultimately raise our money that way, and that's how we keep our lights on.
Talent. Talent is finite. We think it's infinite, but it's not really. We get talent from all over. In medicine and science, we get a lot of talent from the immigrant communities. I think that there's often a shortage of talent. So happens there's a shortage of talent coming into journalism because the economics are terrible. There's a shortage of talent maybe in politics because the scrutiny is awful. It's another subject. How do you feel? Will COVID have an unintended consequence that it will stimulate people not to become just doctors and clinicians, but to become what you are, a research scientist, somebody who looks into the very basis of things and how they are and why they are?
I hope so. One of the most pleasurable activities I've had outside of my own science in my career was when I was on the Johns Hopkins University School of Medicine admission committee. And as I would interview the incoming med students, I noticed year after year they all, and I mean all, had a story that was personal that drove them into medicine. A sick relative, a sick sibling, somebody close to them that died, something that happened in their life that made them turn on this way. And I'm hoping that something similar happens now with young people and science. This tiny little virus is wreaking havoc globally. We can't even go out for dinner because of this virus. Hopefully young people will be turned on to the idea that they can make a difference by going into science and actually sleuthing, doing the detective work to make it better.
Are you developing an exosome map, a green map?
So we're working in conjunction with Dr. Stephen Gould at Johns Hopkins University. He's been a world leader in the development of exosome-based science, really since exosomes were discovered 20 years ago. To work with Steve is an ultimate pleasure because there's very little that he hasn't thought of or worked on in the exosome space. Mapping what's inside an exosome is interesting if you're going to use that as your therapeutic, and we do. We've done that with our exosomes made by our cells. But really what we're doing now is custom loading the exosome. So we're taking what we want to be carried in the exosome. In this case, it would be, for instance, the message for COVID-19 to build a vaccine, but we can put almost anything inside there to derive a biologic response. And so we're less interested in what's already there and more interested in what we're putting in there.
Interesting. Finally, are we going to come out of all this with a much greater reverence for science? Are we going to take experts seriously? Are we going to stop follies like the anti-vaccination movement and other really hard to understand social forces?
Well, it's really interesting. The popular press is so filled with information right now that's so wrong. And I think a lot of the stress that we feel as a society is we just don't know what to believe. And I think social policy has not helped us. And I will say from a top-down approach, our administration hasn't helped us. We've been in wars in this country where our president couldn't stop the war, but he could at least make us feel like somebody was thinking about how to fight the war. We are on a worldwide war against this little virus, and we will ultimately win. How that happens and when that happens is yet to be determined. In terms of driving people to be more curious about science, for me, it's a life passion, so I can't imagine being a 10-year-old girl or young boy and waking up this morning and not thinking how to make the world a better place and help with the opportunity to build science. So I know in my own family, my 5-year-old granddaughter almost every day says, "Grandma, when are you going to get the shot so I can go back to school?"
Well, I think we'll leave it there. I want to tell you, Linda, that I've been interviewing people for decades, too many decades, and you are one of the most extraordinarily articulate interviews I've ever conducted.
Oh, that makes me feel so good. Thank you. It's been a busy week, and so
quite remarkable. Excellent. Thank you so much for coming on the broadcast.